Okay, so welcome to this next video in which we are discussing the opioid receptors. Okay, so we now want to discuss why opioids such as morphine have such powerful analgesic effects. To do this, we need to look at the ascending pain pathways, i.e. how the signal of pain gets up to the brain. We also need to look at the descending pathways which regulate how pain passes up to the brain, basically. Because all of this is going to be important in how opioids take their effect. Okay, right, so we'll start off with how a painful stimulus in the periphery is going to be communicated up to the brain. Okay, so let's say we have our site of our site of pain, basically. Okay, so let's say we have some sort of tissue damage. So maybe we have hurt ourselves and we've got some large bruise developing. Okay, so we've got our site of pain here. Okay, so we'll just call this the site of pain. Okay, and basically many different molecules will be being produced here, and many of them will act on neurons within that area which transmit painful signals up to the brain. Okay, so you will have neurons in this area which have their endings in this area which will be activated by molecules that have been released at this site of pain. Okay, now there are two main types of pain neurons. Okay, there are A delta fibers and there are also C fibers. Okay, now A delta fibers are generally bigger than C fibers and they're generally myelinated. So let's show both of them. So we'll have our C fiber here. Okay, now it's unmyelinated, so I'll have to now show the A delta fiber being myelinated. Now what does it mean to be myelinated? Well, it means that the conduction of the action potential will be much faster, basically. Okay, because the action potential will be able to conduct through these portions of myelin just by diffusion of the sodium ions from one node of RAM-VA to the other, rather than having to go through saltatory conduction basically all the way. Okay, so here comes this A delta fiber. So it will transmit its signals slightly faster than the C fibers, which are unmyelinated. Okay, so both of these types of pain fiber will be activated by molecular signals within the site of pain. Now, basically, we can split the perception of pain, the pain experience, into two portions, basically. We can split it into a first portion, okay, called first pain or primary pain, which is the portion where you've just hurt yourself, maybe you've just cut yourself, and you get a very sharp pain, okay? And this doesn't last that long, okay? So this is the sharp pain. And this is occurring because all of the A delta fibers are now firing, okay? And the A delta fibers firing sends the signal to the brain of the sharp pain. That causes the experience of sharp pain, okay? Then we have this secondary pain or second pain. And this is much duller. This is the sort of achy, often sort of burning pain that that lasts for a much longer time, basically, and lasts much far beyond the original damage that you did. Okay, and this is to tell the brain to not use the affected area, basically, to keep the affected area safe from damage, basically. And this is because the C fibers are still firing. So basically, early on, when you've just damaged this tissue, you'll get the A delta fibers firing like mad, and those will create a very sharp pain up to the brain. Whereas in the aftermath, basically the C fibers will be firing, and those will create the dull painful experience that's secondary pain, basically. Okay, so let's look at how both of these fibers then go into the spinal cord. Okay, and from here on now, we'll just sort of look at one basically, so we won't look at both because the pathway is exactly the same for both. So we'll continue on the A delta fiber basically. Okay, so the A delta fiber will come in through some sort of mixed spinal nerve, so let's say this is a mixed spinal nerve, okay, and then it will be split into the mixed spinal nerve will split into the ventral root, let's say this is this one, okay, and also the dorsal root. So let's show these. So this is the ventral root, and this is the dorsal root, okay, and basically spinal nerves which emerge from the spinal cord. This is the mixed spinal nerve here. Basically, they are formed by the joining of the dorsal root here with the ventral root, basically. Okay, and the dorsal root carries the sensory fibers which are coming in, and the ventral root carries the motor fibers which are coming out. So this is the mixed spinal nerve here. Okay, right. And then what will happen is in the dorsal root you have a special sort of swelling, which is the dorsal root ganglion, before it actually goes into the spinal cord. And then we'll have the ventral root coming out of the spinal cord here. Okay, so I'll show the spinal cord now. So spinal cord's a sort of oval shape, like so. We'll have the ventral sulcus here. And I'll try and make it as symmetrical as possible, like so. Okay, right. So this is our picture of the spinal cord. So let me now label everything up. So this swelling on the dorsal root, this is what's known as the dorsal root ganglion. And because I'm running out of space, I'll just abbreviate it to DRG and write it over here. It's called the dorsal because it's on the dorsal root, and then root, as I say, because it's on the dorsal root, and then ganglion. A ganglion is the name for a collection of neuronal cell bodies that is outside of the central nervous system. Okay, so when you have a collection of cell bodies that is either within the spinal cord or within the brain, it's not called a ganglion, it's called a nucleus. But when you have a collection of cell bodies that's outside of the spinal cord or the brain, that's called a ganglion. Okay? So, all of the sensory neurons that are coming in through the mixed spinal nerve here, will go down the dorsal root. So here comes our A delta fiber down the dorsal root, and basically it will have its cell body within the dorsal root ganglion here, and then it will send a central process into the spinal cord here. Okay, right. So now let's have a look at the anatomy of the spinal cord. So basically, the spinal cord can be divided into white matter and grey matter. Now, grey matter is where you've got neuronal cell bodies, and white matter is where you have the axons. Okay, so let's show this. So, the grey matter is shaped in a sort of butterfly-like shape, okay? So it's like so. Okay, and basically, the posterior extensions of this grey matter, so this portion here and this portion here, are called the dorsal horns, okay? So I'll label this up on this side. So this is a dorsal horn here, okay? And this here, this is the ventral horn. Okay, so you have a dorsal horn and a ventral horn, and you have a ventral horn here and a dorsal horn here as well. Okay, right. So what's going to happen is these central processes coming in from the A delta neurons or the C fibers, those are going to come in and synapse on another neuron that is going to be within this grey matter. Now, the grey matter is divided up into certain portions. So firstly, right at the bottom, you actually don't have a portion of grey matter. That's the first thing to talk about. The grey matter actually starts just after a little portion here. So I just want to firstly talk about this little portion here, okay, which I'm colouring now in pink. Okay, so this little portion in pink is known as Lizauer's tract, or the zone of Lizauer. Okay, but we'll call it Lizauer's tract. Okay, and basically what happens is the central process of the A delta neuron is coming in from the dorsal root. Okay, what will now happen is it will split up. Okay, and it's better to show this looking from a different angle. At the moment we're looking at a cross section. I want to now show you a picture where we're looking from the front. Okay, so we can actually see the length of the spinal cord. Okay, so we were looking at a cross section. Now I'm showing you the spinal cord as though we're just looking at it from the front. Okay, right. So in comes this dorsal root here. Okay, so there's the dorsal root ganglion, let's say, and it's coming in, and here we'll have the ventral root as well. Basically, what happens is as this central process of this A delta neuron comes in, it will split into many different portions, and these can go up to different levels, and they can enter the spinal cord at different levels. So basically, each pain neuron, whether it's an A delta fiber or a C fiber, as it enters the spinal cord, it will split into many different processes, basically. So it splits into different processes, and these different processes will either go up or down or maybe stay at the same level of the spinal cord, so that you're getting input from this neuron at many different levels, basically. Okay, now where do all of these processes go up and down? Well, that's within the Zauer's tract. Okay, so this is all occurring within the Zauer's tract, and of course you have two of them, one on both sides, and they're at the back, basically, they're over here. Okay, behind the dorsal horn. Okay, so unfortunately I've got that arrow of the dorsal horn pointing to the Zauer's tract. This is the beginning of the dorsal horn here. So, basically the grey matter, okay, of the spinal cord can be divided into certain areas, okay, certain laminae as they're called, and these laminae are known as Rexed's laminae. So we're now going to discuss the division of the grey matter of the spinal cord into Rexed's laminae. Okay, so basically right at the centre of the spinal cord, just to start us off, here is the central canal. Okay, so that's got cerebrospinal fluid running down it, and it's the continuation of the fourth ventricle, basically. Okay, in fact, the fourth ventricle is obviously the continuation of the third, and then the first and the second ventricle, so it's the continuation of the ventricular system. Okay, so this is the central canal. Okay, so it's just full of a little fluid, it's not got neurons within it. Okay, so what we're now going to do is look at the division of the grey matter of the spinal cord into these Rexed laminae. Okay, so there are 10 separate Rexed laminae. So the first 6 are within the dorsal horn. So there's 4, 5, and 6. Okay, so these are the first 6 Rexed laminae. So this one right at the back here is Rexed laminae 1. The second one is Rexed laminae 2, and then it goes on in the sort of logical order. So I'm just going to highlight them up just to sort of emphasize the point. Okay, and I should have drawn this picture bigger. This is Rexed laminae 1 that I've just coloured in blue, next to the Zauer's tract. Rexed laminae 2 is in orange, and this is working quite well. I might continue it on. Okay, Rexed laminae 3 is in turquoise. Okay, now I'll put the colour coding down here. So we've got 1 in blue, so let me colour code that in blue. Okay, we've got 2 in orange, and I should just stress that the same is happening on the other side. Okay, I'm only going to do one side, so you have Rexed laminae on both sides. You'll have 1 to 10 for the right-hand side, which we're drawing, the patient's right-hand side, and 1 for the left-hand side as well, because I'm assuming we're looking at this from above, basically. Okay, so then we have Rexed laminae 2, which is colour-coded orange. Rexed laminae 3 was coloured in turquoise. Okay, then we continue on. So Rexed laminae 4, I'm not going to do it in vivid purple, I need more colours. Okay, we'll have it in green here. Okay, and that one's gone a little bit wrong, it's overflowed into Rexed laminae 5, but never mind. We'll just have to pick some strong colour for Rexed laminae 5. Okay, so that's Rexed laminae 4. And generally Rexed laminae are named with Latin numbers, just to be pretentious. Oh, sorry, Roman numerals, rather. Okay, and then here is rexidlamine 5 here, okay, in red, like so. And then finally, rexidlamine 6 is in, I'll do it in vivid purple, is this one here. Okay, so that's rexidlamine 1, 2, 3, 4, 5, and then finally 6 here. Okay, now let's go on. So the rest of the grey matter on this right-hand side still has to be divided up. So first I'm just going to chop it in half. So we're not going to touch the left-hand side. We're only going to do rexidlamines on the right-hand side. So firstly, let's show rexidlamine 10, because that's quite a nice simple one to do. Basically, rexidlamine 10 is the portion that faces the other side of the grey matter. So this portion here is rexidlamine 10. Okay, so I'll put this here. And then what colour should I colour that one in? I'll do it in yellow. Okay, so this is rexidlamine 10. Okay, then we'll show rexidlamine 8, and then rexidlamine 9. And then basically all that's left is rexidlamine 7. Okay, so next up is rexidlamine 8, which goes in here. Okay, and we'll put this in pink, I think. Okay, so this is rexidlamine 8, like so. So 8 will fit in here. So that's rexidlamine 8. And now we want rexidlamine 9. Rexidlamine 9 is split into 3 little portions. 1, 2, 3, like so. And we'll colour those in turquoise again. I'm going to have to start using the same colours again because I've run out. Okay, so this is rexidlamine 9. What's the Roman numeral for 9? Like this. Okay, and then finally we have rexidlamine 7, which is everything left over. So I won't try and colour that in because it will just ruin the picture. So all of this portion here is then rexidlamine 7. Right, so those are the ways that the grey matter of the spinal cord is divided up into rexidlamine. Okay, so what was the point of discussing this? Well, basically the processes that are coming in from the A delta fibre are coming into the rexidlamine, and they're going to synapse in the rexidlamine. Now, which rexidlamine are they going to synapse in? Well, they're going to synapse within the rexidlamine 1 and the rexidlamine 2, which are collectively called the substantia gelatinosa. Okay, so rexidlamine 1 and 2 collectively are called substantia gelatinosa. Substantia gelatinosa. Okay, and we'll continue this discussion in the next video.